Hi everyone, my name is Oleksandra I'm a second year PhD student at Rouhanifard Lab.
Hi, I'm Kequing. I'm fifth year PhD student in the Rouhanifard Lab.
In our lab, we do studies about RNA modifications and RNA in general, which is the molecule that helps our body make proteins. My role is to use very fancy equipment as Oxford nanopore sequencers to sequence RNA and study and try to characterize RNA modifications.
So my goal is to develop a new technology that we can use to study the RNA dynamics because we know because the live cells, the gene expression is dynamic. It can be like here or there or the amount of the RNAs could be like different. So like we want to track the RNA expression in live cells.
What we try to do is to study RNA modifications and understand why they are in human body and what is their role in our bodies. Pseudouridine recently became a very popular topic in research because it has been used for COVID-19 vaccine development, and last year's Nobel Prize for medicine or physiology was awarded for pseudouridine. So pseudouridine is one of the RNA mods we are looking into, and our goal is to characterize it and find maybe new functions and try to maybe solve new diseases or develop some new methods to help RNA-based medicine in the future.
My project is kind of because we know the RNA expression or the gene expression is dynamic. People usually do they just treat the cells and then wait for several hours, then kill the cells, then do the kind of gene expression studies. It's kind of a dynamic sense, but it's not that like really dynamic because it's not in live cells. So my project is kind of want to develop a new technology that we can use to fully understanding and to studying the gene expression, the RNA expression, so that we can control the gene expression or like we can drive the therapeutic advances. We kind of want to develop like a kind of reagent that we can use to put them on the cells so that there can be a click chemistry reaction happen there. So we can know, oh, there's like RNAs there, or there's kind of protein there, or there's kind of like any other major biomolecules in the cells like lipid, sugar, or DNA for now. Yeah.
I like being in Rouhanifard Lab because what we do is very new. We use cutting edge technologies every day, and we try to solve problems that very few people around the world are working on. So it's very challenging every day, that's true, but it's also very fun because it's new. No one else has done this before. So that's why I really like being here, because this allows me to work with other people that are expert in this field, and this kind of prepares me to whatever challenges will come next in the future.
For me, my work is most about the bench work. It's wet lab, but also in Rouhanifard Lab we can, because when we get the data, like we need to analyze the data. So that means we need to use some coding like MATLAB or R, which is fun because I never do the coding things before in the college. So I kind of think it's gonna be really, you learn a lot of stuff, you learn a lot of skills, which is really helpful. And also in Rouhanifard Lab, we are open to like master students or undergraduate students. They'll come to our lab. They want to learn. So we are really want to like teach them like the skills we have, the experience, the knowledge you have. We're sharing the knowledge. So like it's kind of a communication skills you build up.
As far as changes in my like perspective. I started this research during my PhD, so this was very new to me, but it's very fun and challenging, so I really like it. So my perspective really changed during this PhD because I really started to do new things that I never did before.
Yeah, for me, I also think it's kind of fun because before I came to this lab, I never did the tissue culture or cell culture experiment before. So it's really fun, you know. Every day you see the cells, and then the other day you will see all the cells. They are all grew. It's gonna be fun. And also for my project, it's kind of tough and challenging. I spent like for 4 years in this project because it's kind of like life systems, so it's not that easy. And like for my like when I develop the reagents to apply in the cells, the reagents we need to purify it so that it can be like not toxic to cells. And then during the purification process, it's gonna be hard because I tried a lot of methods we can use to purify the reagents, but they were not working very well. So like one day I talked with my ex roommates about my trouble, and I cannot solve this problem. She like gave me a suggestions, and at that moment I went, oh my god, I can use this kind of method to purify my reagents. It's gonna be really, it works. It did works. And then I tried like half a years to kind of solve this problem. And I never realized that talking with people, not like search papers or like talk with your PI, can really help you to solve some problems that you never experience. Yeah.